Hi, I'm Barry Honig. I gather I'm chairing this session, so I'll introduce myself. Again, I'm Barry Honig. I'm a professor at Columbia in the departments of system biology and biochemistry and molecular biophysics. And one of the things we do in our lab, we're trying to do, is to integrate structural biology with systems biology in a number of ways. And I'd say a key element of the way we go about things is to use protein structure the way many people use sequence. And you'll see an example of that. But the notion is that if proteins have similar structures or similar parts of structures, then that may inform us about function just the way sequence similarity can inform us about function. And for the purposes of this talk and a lot of what we do, I'll introduce our PrEPI program. Have to stand here. PrEPI, it's not going to work. PrEPI starts with two proteins. In this case, you see that in yellow and green. And if there are structures for those proteins, either from the protein data bank or from homology models shown here, it then looks for proteins of similar structure, which we call structural neighbors, and the yellow protein has these neighbors, the green protein has these neighbors. If any of these these proteins, and there's about an average, we find 1,000 of each. So if any of one of the million pairs of possible interactions is a known structure in the protein data bank, we have a model of that complex. We superimpose our proteins on those proteins, our unknown proteins on the known proteins, and we get a model of a complex which we then score with a certain likelihood that it's going to form a complex. I'm not going to be talking about the details. I just want to give you a sense of what it is we do. How do I advance the slides? Is there, with my computer, maybe? No? It's so reassuring to know it's not my fault. Okay, thank you. Great. So just to give you a sense of how this might work, these are, again, the yellow and green proteins. We actually built a model for how these proteins interact based on, oops, now it's hyperactive. Based on these two guys and these structures. Oh, see, it's misbehaving. The chairman may give me an extra minute or two to get this resolved. Anyway, the two proteins here are from Arabidopsis. We're superimposing our human models or structures on the Arabidopsis proteins to get this putative complex and scoring it. So we're using structure in this way. We're using structure, again, as I said, the way people use sequence. We integrate structural clues with non-structural clues, co-expression, whether they're known to have related, the proteins are known to have related proteins, functions, and we eventually come up with a database we call the PrEPI database, and PrEPI looks something like this. You can access it. You can get sources of information. You can get models of the structures we predict, map SNPs on them or cancer mutations, and get information. The main point to make is, there's a missing slide. No, here it is. That because we have lots of structures, and because we've sort of learned to work genome-wide, PrEP is very large. It has over 1 million predicted interactions with what we call a reasonable, probability of being correct based on some likelihood ratio. Of those, of the 1.35 million, 1.1 appear nowhere else. And we believe that about a quarter of those, perhaps more, are actual physical interactions. So not all PrEP interactions are necessarily physical because we use non-structural sources of evidence. So it's a very, very large database. We've tested it in any number of ways, and it seems to perform quite well. So we have a number of applications that we've published and others that are not published. This is a result that is published. We looked at proteins in the ClinVar database and looked at pairs of proteins that are associated with the same disease, and ask if PrEP predicts that they interact, and then ask the same question if they're associated with different disease or just choosing proteins randomly. And I think you can see here that we get a very large enrichment. Proteins associated with the same disease are predicted by PrEP to interact. What I'm not showing here, because I'm sort of just giving you bits of what we've done, is that there's a very high enrichment of disease mutations in PrEP predicted interfaces. So what we have, again, is a very large database with structures and with the possibility of predicting whether particular mutations are in interfaces or not. Now, we've used that information to try to predict whether, say, cancer mutations or whether proteins associated with cancer mutations are likely to, whether we can predict cancer mutations. And we've done this in the following way. We start with a PrEP network, and here's the way we define these networks. Here's a protein P, and we look at neighboring interactions. So we have a PrEP interactome, and everything shaded here in yellow is generated with a random walk with restart algorithms. So everything in yellow we think of as being in the neighborhood of protein P. And then for cancer mutations, we map on this network a number of properties. One is, does a mutation disrupt an interface formed by protein P? And you see here there's 2 of those, so this should say 2, I can't see. Are there disruptions of interactions in the PrEP neighborhood, in the protein neighborhood? We count those, and then we count the disruption of interactions of proteins shown here in yellow with the same function as P. So we do that for cancer-associated mutations. We do that for germline mutations. We combine it all in a random forest algorithm and ultimately come up with predictions whether a particular protein is associated with cancer or not. And in this way, we think we can be getting at some of the problems Trey was alluding to, of not only looking at particular proteins, but whether networks are associated with disease, whether there are mutations that you wouldn't necessarily associate with disease somehow coming out, and our ability so far to make predictions of this sort, that you see we only actually predict one protein that's in a germline mutation is very encouraging for us, and we're continuing in this effort, but it hasn't been published yet, so I'm just sort of giving you a sense of where we're going. Another thing we've done with PrEP is to integrate it with regulatory networks from Andrea Califano's lab. Andrea and I collaborate very closely, so he's come up with programs and networks called Arachne, Viper, and Cindy based on the analysis of tumor samples, and we combine those with PrEP in using random forest. And the notion here is that Andrea's programs come up with directionality, and they're tissue-specific. So he has tissue-specific networks, direction information, and PrEP is more like a canonical pathway, except it's not a pathway, it's a network, and that contains basic, say, physical information combined with Andrea's regulatory networks. And here's an example with KRAS. We have upstream modulators, downstream effectors, and we have a KRAS-specific map or network, as we call it, which combines Andrea's tools and the tools from my lab. We've tested some of our predictions on KRAS with Andrea Sweet-Cordero at Stanford, these experiments are done in organoids associated with lung cancer, and these are predictions. We come up with, get the numbers, 40 or so novel predictions. We also recapitulate some known predictions, and as you can see from this graph, many of our predictions, most of our predictions behave as synthetic lethals with KRAS mutations. So in this test, at least, we're really doing very well. Again, this work is still, it's not published yet, but we're very encouraged by this result as well. This is just an expansion of the KRAS map. And you see here a protein, I don't know how to pronounce it, DYRK1B, which is predicted to interact with KRAS. And the OncoSig program basically predicts an interaction between DYRK1B and KRAS. But if we just run PrEPI alone on the proteins of interest, we come up with a predicted physical interaction between DYRK1B and a protein called calorin. So this allows us to add a physical sort of basis for the connection between DYRK1B and KRAS. And this is work from Diana Murray, who's here in the audience. And here you can see more or less how this is predicted to work. Here's DYRK1B, here's calorin, and they interact through kinase domains, shown here, but then calorin interacts with KRAS through a GEF domain. So what we see here is the beginnings of a structural network, which again, PrEPI is providing for us, and we're adding information that you don't get from the direct OncoSig predictions for KRAS interactions. We're using PrEPI to look into and to provide a structural basis for many of the networks that everybody uses, and this is the mTOR pathway. And now if we add PrEPI-based predictions to that, some of the known interactions are recapitulated. In addition, we're predicting a new set of physical interactions in that pathway and in related pathways. And I think it's interesting to get a picture of how we're predicting that this occurs. There's a whole series of different interaction domains that are playing a role in this pathway. Two SH2 domains, GRAB2, dead domain interacting with a kinase domain, et cetera, et cetera. We don't know whether these proteins are actually in multi-protein complexes and perhaps interacting with sort of the Bioplex database might be a way to get at those questions. But in each case, at least we have a structural model that can be tested in any number of ways. We're also using, we have what we call a drug PrEPI, which uses the same principles to predict whether a small molecule interacts with different proteins, again on a genome-wide scale. And we're trying to begin to use that for phenotypic screening, but also to find ways to think about how to use drugs to drug entire networks rather than just individual proteins. And of course, we're not the only groups that are interested in that. So that's sort of an overview of what we do. I wanted to make this audience aware of the power of structure, of the possible connections it can make. And I left enough time for questions. I just want to mention the names of the people that did the work. Donald Petrie and Diana Murray are senior scientists in the lab. Cliff Zhang designed the first version of PrEPI, and Jose Garzón the second. Josh Broida worked on OncoSig. And Gorka Lasso worked with Sagi Shapiro at Columbia to actually come up with a PrEPI for virus-host interactions. So far, we've only done it for human and for yeast, but there's no reason not to use structural information to make predictions about viral host interactions. And that's it. Thanks for your attention.
Hey, Barry, great talk. One question, are you thinking, or how are you thinking about incorporating post-translational modification information in to your structural analysis? Because of course a lot of PPIs are mediated by PTMs.
Thinking about it, it's not easy, in part because our representation of proteins is quite crude, in the sense that we're just looking at secondary structure elements. Where specific amino acids come into play is in the interface. So if we know where there are modifications, then we can use that to score an interaction, and perhaps in one case it'll form, in one case after something is phosphorylated it won't form. My sense is, in our first run through this, since we want to do something on a genome-wide scale, as they say, we have 1 million interactions, we have predictions for I think about 18,000 proteins. To get that scale, we had to sacrifice a lot. And since it's a hypothesis generating program, the hope is that the next stage, if there are interesting hypotheses, we use different filters. And at that point, I think it's a place to introduce post-translational modifications.
So I guess following on what you were just saying, if you were to take this beautiful network of interactions and filter it by cell type expression so that you only looked at the proteins that were expressed together in the same cell types, have you thought about that, or does that work?
So it's easy to say to everybody, yes, I've thought about that as well.
Yeah, obviously.
But I think, again, the goal, so we're at a stage, I have to say, putting this together was a big job. We're dealing with actually considering billions of possible interactions using diverse sources of data. We have it now, and the next step is beyond just the database itself, is to introduce filters of various sorts, to have PrEP for this, PrEP for that, and the work with Andrea was a tissue-specific and a cancer-specific PrEP, but I recognize that if we can find a way, perhaps we need resources to do exactly what you're asking, and I hope if we write a grant, you'll review it then.
Absolutely.
But that's where we'd like very much to go.